Hi everybody, it's me, Mr. Olson, and I'm going to teach you today about the nature of science. It's our first lesson in biology that we study because knowing what science is so important. So that's what we're going to focus on today. We're going to talk about the scientific method, we're going to talk about experimental design and components or factors of an experiment, and we're also going to talk about theories and laws and even how to design and make your own hypothesis. We'll try to work through a little bit of information, a few scenarios, and we'll try to learn a little bit about science. Here we go. So, what is science? Well, science is basically a way to explain how the universe around us really works. It's done, I think, and two of the most important words here are going to be in a systematic and a repeatable way. Basically meaning there's a system to it. Now, science kind of has two main components. First off, it is considered a way of thinking, a scientific way of thinking, a scientific process. And if you've heard of the scientific method, that's what that is. It's a more of an elaborate version of trial and error where you use data and you really think about what your results are to form conclusions based on your original idea. It's not just a random thought. It's also a body of knowledge, and by that I mean we're actually having a large body of knowledge, a large source of information that confirms our ideas. We know science has discovered quite a bit of stuff, and we consider that part of our understanding. Knowing that stuff is really important. For example, if you were having a conversation with a friend and they said, "Oh, what's the hardest object in the world, or hardest object you can find?" It would be a diamond, and you could simply reply, "Oh, that's science. I know that because of science." And the other thing is science is supported with something called empirical evidence. Now, empirical evidence basically means it's evidence that we can measure, it's tangible, it's something that we can actually have. It's not just some idea, it's not fantastical, it's not just anecdotal information, it's literally something we can actually hold, touch, measure, observe. Without that, it's hard to confirm your thoughts and your ideas. I'm not saying it's impossible. Some experiments have been done as just thoughts, and we've learned a lot from them, and we've grown quite a bit using them. Good examples of a science would be a biology, which is the study of life. We've got physics and chemistry, and really physics kind of runs the show. They can explain a whole lot of things using physics. And astronomy is a good example. As you guys know, we have the eclipse happening very soon. And yeah, basically we're saying we have an understanding of the stars, of the planets, and all the other celestial bodies that interact. I'm sure you can think of other examples of what might constitute science, but let's see if we can compare that to something else. A good example of something that would be a non-science would be something like philosophy or technology or literature, even religion. In no way am I poo-pooing any of these ideas. As a matter of fact, these are all pretty great things. We're just simply saying a non-science is something that's not a science. So these play a role in the world, but they don't play a role in science. What we're trying to do is work with empirical evidence. These other things don't really lend themselves to that. A lot of the times, a lot of these topics are considered pretty subjective as opposed to objective. Subjective is more biased or more opinionated, whereas objective is more rooted in real evidence, and it can't be really debated. And that's what we're trying to avoid in science. We want a reliable and understandable way for everybody to agree on some conclusions. Another, I guess, kind of science that's out there is a pseudoscience. Now, the word pseudo would actually mean the word fake, which simply means that a fake science is something that purports itself to be a science, but there actually isn't any evidence to support their claims. This usually causes a lot of confusion in the public. In a lot of cases, we see people who will try to sell things to others saying, "Oh, it's scientifically proven," but they might even claim they have studies. But if you look at their studies, they don't really use a method in any reliable way. They're not using a system necessarily. They may be lacking controls in their experimental design, and we'll get to those here in a second. And they're purporting things, or they're saying things that can't really be justified. And that's kind of dangerous. Things like astrology, people might believe that astrology is important because they can look at star charts and measure things, but that doesn't actually mean it's scientifically rooted. Since you know where Mercury and Venus are and you know what time of the year you were born, it does not tell you your odds of being lucky that time of the year. Also, homeopathic medicine or homeopathy and essential oils, these have been growing in trends in recent years, and there's not actual documented medical evidence that suggests these things do anything. Now, if you like them, if you enjoy them, and if you think that makes you feel better, more power to you. But don't walk around thinking this is a good substitute for medical advice and science. It's not, and a lot of the times you can be taken advantage of financially by following these measures. So when it comes to evidence, there are two main types of evidence. The first type is going to be qualitative, which is kind of familiar to all of us. It really depends on our senses. Things like how something smells or how does it taste, if it tastes salty or sweet. You could describe that to a person and they'd understand it, and that would be considered qualitative information. We also have color. Now, I know in like a computer-based sense, you can assign a number to a color, but otherwise, in real life, you probably can't. Somebody might look at a color and call it sky blue. Some people might call it baby blue. Some people might say it's light blue. And it's going to be a bit subjective. A lot of these are a bit subjective, but they're still important to us in science because it can at least help you describe an event that's occurring, or at least making an observation. When it comes to quantitative, this is more of that empirical evidence that we're looking for. It's a measurable number to some degree, or some kind of data point or datum. We have temperature, and in science we don't really try to use most of the American based units. We try to use, in biology at least, we try to use Celsius whenever possible. And that's simply because water freezes at 0 degrees Celsius and boils at 100 degrees Celsius. And it makes it a little bit easier to calculate when and where we're going to find life in those conditions. We usually don't deal with life well below freezing or near boiling. When it comes to measuring distance, we usually use centimeters or possibly meters. If you're going with large distances or sizes of things, you could say kilometers. And in biology, we get to the micro scale too. So we use micrometers, which is like a funny looking U, and we have nanometers, and we get really, really small. But the other thing that we try to focus on is going to be mass and weight. Now, just keep in mind, mass and weight are different from each other. Mass is basically going to be how much matter, how much stuff is packed into an object, whereas weight is going to account for mass, but it's going to be based on gravity. So mass is really what we try to use all the time if we can. We use triple beam balances to measure those all the time here in class. And we usually use either kilograms or, in general, grams. If we're working with small numbers, it could be milligrams. And we use all kinds of other units too. But yeah, so you should be familiar with those forms of quantitative evidence. When it comes to a scientific design, we need to have a hypothesis in place. Now, I know you guys have seen that word or heard that word or even used that word probably plenty of times in your life, and especially here in school. But we're going to define a hypothesis beyond just what most people define it as. A lot of the times people just say a hypothesis is an educated guess, which is well intended, but it's not exactly what we're looking for. A hypothesis, I think, is more appropriately labeled as a testable prediction for explaining some sort of phenomenon or phenomena that we can observe. Now, it's designed to be tested with some sort of experiment or observation in mind. Now, one note here, I kind of want to make a little arrow here for you. Your hypothesis is technically never really proven. In science, we don't work on proving anything. As a matter of fact, most of the best ideas we've ever thought up of are the best ideas because they've survived such an ongoing onslaught of people trying to tear it down. And if we just keep finding evidence that supports it, we just keep saying we failed to reject it. It's like saying accepted, but if we accept something and then we get more information later on, then we look kind of silly, like we've got a little pie on our face. So we never we don't really accept it, we just basically fail to reject it, or we outright reject a claim. And it's usually written, at least in our level, not at a higher academic level, they don't usually follow this little rule, but we will be writing them mostly as if-then statements. So if blank, then blank statements. And we'll play a role about, we'll talk a little bit later about exactly what you want to write in those blanks. It depends on the scenario, and we have a whole assignment and activity created for us to be able to do that. And also, the hypothesis should guide all of our experimental design, and that's what we want to get to next, is talking about how to design an actual experiment. So, like I was mentioning earlier, whenever we want to test an idea in science, in a field of science, we want to use a scientific method, or the scientific method. The basic flow of ideas basically says you come up with an idea, an original thought that you would like to have tested. You take that idea and you kind of transform it into a hypothesis. From that hypothesis, you put that into an experiment, you work through it, you test that idea, and then you collect data. And then from that data, you support your claim, which was your original hypothesis, and you see if your idea worked out the way you expected it to. Let's go ahead and make me draw terribly, but we're going to draw what that scientific method looks like. So, going to start at the top here. We are going to go ahead and say the first thing you may notice in coming up with an experiment is that you either start off with a problem, or you can even start off with just a generalized question. And this is a good basis to just kind of wonder about the world. You know, you notice something, you want to know why or what's happening, and maybe we can figure out. So after this, we're going to kind of work in a circle here. We basically have to generate a hypothesis, a hypothesis. A hypothesis should not just come out of the blue. So I just want to go ahead and kind of draw outside of this circle a smaller one, and we should have ongoing research that guides our design here. This is really a ceaseless project. You should be doing this all the time. After you come up with a problem or a question, most people, at least reasonable people, will probably go out and see if it's been answered before, or get some information about it if it's not been answered before, and see if that can support some of their thoughts. We shouldn't just randomly come up with a hypothesis. We should definitely think about it and research beforehand. Say, for instance, you were doing cancer research, and you have a new kind of chemical or a new kind of drug that you'd like to see if it worked on cancer cells. Well, if you didn't do research and you ran this experiment and then later found out somebody actually had already developed the same thing and got bad results, then it was kind of fruitless and not really worth your effort. So it would be good to do research and maybe tweak your ideas as you can. From a hypothesis, we're going to go ahead and go through a process where we design an experiment. And I'll just say EXP. And then after you design it, I know it seems a little redundant, but you need to actually do the experiment. And we'll talk about what experimental design is made up of here after this. But when you design your experiment, you actually have to follow through and do the one you designed. Why? Because your design was based on your hypothesis. A lot of students will make the mistake, especially if I ever make you do an inquiry lab. I give you materials, and then you have to go out there and build it and do it yourself. I usually have to approve of whatever your materials are and what your procedure will be, just to make sure you're not going to do anything crazy. If you plan to do something and then you come in and you do something totally different, you may not be testing your actual hypothesis, and that's going to cause problems when it comes to making a claim. So once you do your experiment, you move forward and you collect data, as I mentioned. And then from that data, which, by the way, we have the two Qs, qualitative and quantitative types of data, we can go ahead and we can form a conclusion. Usually you analyze your data, try to make sense of it, notice trends, and try to incorporate that into an explanation that kind of says, here's my hypothesis, this is what was right, this is what was wrong, this is why, and the data really shows this is true because of this reason. So that kind of brings us back to the beginning when we are forming our conclusions. We always want to base it back on the hypothesis. Now, if we by any chance test a hypothesis and form a conclusion that says, no, I don't think my hypothesis is correct, then we'd say we simply reject the hypothesis and we give up. Okay, we don't give up. As a matter of fact, we do just the opposite. It actually should be a sign of encouragement to you. It actually should promote you to do more. So basically the idea is that we go ahead and we revise and we edit, or we change some portions of our experimental design, and we try again. In a lot of cases, maybe if you have a bad hypothesis, maybe you change the hypothesis. There could be situations where maybe your hypothesis is okay, but you just designed something wrong. It actually wasn't even testing your hypothesis, or something was a little bit off. Well, this is your chance to go back and redesign it. There's even a good, strong possibility, since we're humans, we do make mistakes. When you did the experiment, you did it wrong. Something wasn't measured correctly. You forgot to measure it. Something along those lines. Human error can account for a good number of mistakes in the world, and especially in the scientific community. But let's go ahead and say we fail to reject. I'm going to say accept in little quotes there, because we're not really accepting, but you're failing to reject. If we accept, we actually don't stop. We don't just go, yay, I did it, I'm right, and then we just quit. We actually have to move on to a process called peer review. Now, peer review is basically the idea that you can have fellow colleagues, people that are in your field that know what they're talking about and know what you're talking about. They read your work, they see your procedures, they see your methods, and they say, oh, I see how they reach this conclusion, and I would form the same one. Or if they're more able, they might be able to run your experiment themselves and see if they can replicate it. If they can end up replicating your results, well, you might have a really good, strong idea on your hands that could get tested over and over and over again, might spur new kind of research, new kinds of experiments, and you might see a combination of or collaboration between multiple scientists forming a new idea, which could promote a new source of knowledge that is accepted as a fact in science and is part of the body of knowledge. So that's roughly the process of the scientific method. I know a lot of people might make it look a little bit different from time to time, but that's how I'm going to use it, because I think that hits some of those major things. So when we're talking about that experimental design, there are four main components of actually designing the experiment after we make a hypothesis that we want to identify. The first is going to be the independent variable. You should know what you're trying to test. Sometimes the independent variable is called the manipulated variable in some circles, like maybe on your EOC they may reference it that way. And basically it's the thing that we are tinkering around with, we're modifying, and we're going to see if this causes an expected result, if this can maybe cause the thing we're looking to see. And that's what we're really trying to ask ourselves. When it comes to a dependent variable, what we're trying to do is we're trying to find a factor of the experiment that we're trying to measure, the results, the measured results. Sometimes this is even called the measured variable. This is going to be the thing that you always have to have in place, because we can't determine success without this. We also have constants, or constant variables. These are factors of the experiment that remain the same in all the different test groups you do, or at all the different trials that you do. And a lot of times you don't just do one experiment. You try to do as many samples as you can to increase the statistical average, so you can actually find out if what you're saying is correct. You can't just do things once. You should do them multiple times. So a constant is going to be consistent throughout all of the tests. If, let's say, we were going to design a new kind of heart medication to lower blood pressure, right? And I wanted to test it to people. Well, I'd find people of roughly the same health level. Maybe they all have high blood pressure, and I want to see if I can drop it. We can apply this drug, and we can give it to people in one type of way. We can give it orally. We can make them take it with food. We can make it take them at lunchtime in the middle of the day. And we can make sure that they take it with water. We can also make sure that none of these people have adverse health conditions that are not going to respond to this medication. Make sure they don't have allergies or anything like that. We just want to make sure everything is consistent, so we can see if this drug has an effect on the way that we expect it to. By the way, in that example there, the independent variable would be, well, it would be the actual drug, right? What drug are we using to influence the change in heart health? The dependent variable is the thing we're looking for, which would be blood pressure. So we could identify all these different variables pretty easily, hopefully. The control variable, or really a control group, is usually what it's going to be called, is one of the test groups or one of the test samples or a trial run that does not receive the independent variable. Now this is important because this is something that we're eventually going to compare our results from the other test subjects that use the independent variable to. Without a control group, it's hard to verify any change in the conditions and any change in the dependent variable. So it's important to have that. Let's go ahead and say we can, like, for example, try to determine what some of those real life scenarios might be, and we can identify some variables. So I'm going to draw some beautiful cups for you. I'm going to draw 4 of them, and each one of them, they're all supposed to be equally sized, by the way. I know it doesn't look that way, but that's what it's supposed to be. 4 cups, little pots that we're going to fill with soil. And they're going to have the same kind of pH in the soil. They're going to have the same kind of moisture content. They're going to be made of the same kind of stuff. And inside, we're going to plant a seed in every one. It's going to be the same size and same type of seed. Let's say today it's a little daisy. And then we are going to expose them to the same environmental conditions. We're going to put them in, maybe like inside of a laboratory. We're going to put them in a room. The temperature is going to be controlled at all times. It'll be a favorable temperature. It'll be warm, but not too warm. We'll make sure they're exposed to the same type of light 24/7, so there's no nighttime at all. There's no breeze. There's no nothing like that. And everything's going to be the same, except the thing we're testing. We're trying to figure out, does water cause plants to grow or cause daisies to grow? So in one of them, we're going to add 1 drop of water, and the other one we're going to add maybe say 2 drops of water or just a little bit of water. And then in the next one here, we're going to go ahead and add a lot more water than the others. And the last one, we're not going to add any water at all. Now, we can kind of predict what's going to happen here. It doesn't really matter any numbers for this example, but for this first one here, you'd probably expect it to grow a little bit. But the next step up, you would expect it to grow a lot more. And in the final example here, of course it's going to grow a lot more, and it might even grow a little actual flower around it because it's doing so well. In the other example with no water, it might sprout at first, but of course it's eventually going to die depending on how long we run this experiment. It might sprout just a little bit, but beyond that, no. Now, this is important because it helps us identify some information. Now, let's go ahead and say we have the independent variable. We're looking for that. That would be, in this example, the thing we're changing, which would be the amount of water that we're adding to these plants. When it comes to the dependent variable, what we're really looking for is going to be the results, and what we're trying to measure is going to be the growth of the plants. Depending on how much water we give the plants, it will result in growth. When it comes to some constants, well, I already listed a lot for you, but let's go ahead and write them down. We can have information about the amount of light that they receive. We can say the size of the cups. We can say the seeds themselves, they're all the same, they're the same size. We can say the type of water we're adding. We're not adding salt or I don't know, sugar or anything weird. No use of fertilizers. We're saying the temperature was the same. You could probably come up with 12 more things, just kind of based on this silly example, pretty simple one. The last thing we want to identify is going to be the control group, which is very important because once again we are going to compare our results to this, and that would be, in this case, that'd be the cup missing the water because we've removed the independent variable, the use of water. We've taken that away, and we're going to go ahead and measure the results there. Keep in mind, the big thing is it does not have the independent variable present, and that is a dead giveaway to find out what exactly the control group is. So let's say after we go through our experiment, we've gone through our design, we've got our results, and let's say we've gone through peer review because we had a pretty successful hypothesis. Well, over a long time and lots of effort and lots of people looking at your stuff, if they all seem to gravitate towards supporting it, and it's some kind of breakthrough source of information, we can develop something called a theory. Now, theories are not what most people think they are. A lot of the times people just think as a theory is just a good idea. No, a theory is a bit more beyond that. It's not just a theory about what's going to happen on the next episode of Game of Thrones. A theory is going to be literally the most highly supported idea people have ever thought of in terms of science, maybe ever in human history, because it's basically a hypothesis that's been tested over and over and over again, and other ideas just seem to feed into this thing and just support that claim. It also does the job of explaining how or why. Really, it's going to be a how or why a phenomenon occurs. Good examples of this include cell theory. None of you will doubt that you're made of cells. You've probably looked at cells before in a microscope, but it's still considered a cell theory because, well, it's the best idea we have, and it's not been broken apart, and other ideas go to support it, and it explains how and why cells are organized, and how they function, and what components go to make them work the way they do, and how they came to being. So if you don't doubt cell theory, because I don't think many people do, yet a lot of people make a stink about not supporting the theory of evolution. And evolution is easily the most widely supported idea in the field of biology, just based on evidence alone. Everything from cell design, like we mentioned there, to DNA, a fairly newish field, and genetics, all support the idea of evolution taking place. We can look at animal behaviors, we can look at how reproduction takes place. A lot of physical design of things that are alive seem to all fit this idea. So when people just say, oh, evolution, it's just a theory, that just tells me they don't know what a theory really means. It's not a weak idea. It's very, very strong, and it's not easy to just dismiss. Now, I also want to mention a theory can do a unique thing, and that means it can change. Because once again, it explains something, but if we get new information, we can adjust the framework a little bit. I've never really heard of many theories that are just totally dashed apart. But we can change and we can tweak, especially nowadays. We get better technology, we receive more information, and we kind of run through things in a different way than we did 20 or 30 or 40 years ago, which means we can learn a lot more stuff and hopefully put it into action. Now, a law is different than a theory. A lot of people think a law is just a graduated form of a theory, and that's not true. Really, a law describes a phenomenon in terms of what it actually ended up doing. It doesn't explain the why or the what or how, but it explains basically what that activity is, what that phenomenon is. A good example I think everybody should be able to understand is going to be Newton's laws of motion. You know, we all know about gravity, you've heard of it, and that's because, you know, the planet that you're on has a giant pile of pull, of force that brings you closer toward it. And also, this just explains, you know, why things move the way they do. Why does the baseball hit the ground eventually? Well, gravity pulls it to the ground. Why does the baseball slow down too? Well, maybe there's friction from the air because there's stuff in the way that the ball is hitting. If you were to do this in space and throw it, and there's a vacuum, well, the ball's going to continue to float through space in a straight line until whenever. Until something basically stops it from happening, either gravity or it hits a giant space station. I don't know. But a law is just basically a really firm, accepted scientific fact, and theories go on to basically describe how certain things occur.